Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International GCSE Biology Paper 1BR for January 2022. I will do 3 parts of this paper. This is part 1. I will do part 2 as well as part 3. And I will put their links or links to their videos in the description box below this video. Please do not forget to check them out. Let us begin with the first question. Question 1 says, the diagram shows part of a food web for a desert community. So these are desert organisms, as you can see. So they say, how many organisms in this food web are secondary consumers? So if this is the producer, we expect in this specific food chain, or you remember this is a food web, but it has many food chains. So taking this and that, we can see this is a primary consumer, that is a secondary consumer, that is one. And then if this is a producer, this is going to be a primary consumer, again, that is a secondary consumer. So if this is a producer, primary consumer, secondary consumer. So we can see there is only that and that. So we have 2 organisms that are secondary consumers. Let me go to the next part. They say, draw the longest food chain in this food web. The longest food chain is that, to that, to that, to that, and to that, as you can see here. The key thing in drawing a food chain is the arrow has to point to the thing that eats the other. So in this case, the acacia is eaten by the desert rat. That's why we see the arrow points towards the desert rat. And then here, the fennec fox. The desert rat is eaten by the fennec fox, which is the monitor lizard, and so on. So the arrows have to be pointing in the right direction for you to be able to get the mark for that. The next part says, explain why most of the energy in the producers is not transferred to the hog. Again, we can go back in this specific food chain. Energy from the producer, which is the acacia plant, has to be taken to the hog. However, so many processes and losses of energy occur within this area. Those energy losses could be due to some parts of maybe the acacia plant not being eaten. Things like roots may not be eaten by the desert rat, or the bones of the desert rat may not be eaten by the fennec fox. You could say the bones as well as the hair. So many things are going to occur due to energy losses. There is another thing in other energy losses, like in excretion, as well as maybe in energy when respiration takes place. All those things are going to cause losses. So let's see. I say energy loss due to undigested or unabsorbed food. This is in form of feces. Parts of this plant are going to be eaten by this, but they will not be digested, so they're going to remain as feces that is going to be ingested out of the desert rat's body. So the energy they contained is not going to be carried on to the fennec fox, and it will not be carried on to the hog. So that is one way. The other one is energy loss through excretion. Excretion is going to be when things are released, maybe through sweat or through urine. That is a loss. So there is also energy loss due to unconsumed parts like stems and roots. That is for this. Or you could say bones, hair, and skin. There is also energy loss through heat during respiration. Heat lost in respiration is not going to be, that energy is not going to be carried on to the hog. The energy lost due to movement and activity, as these organisms within this chain move along, they use energy, so that will not be transferred to the hog. Let's continue. So this is a photograph shows the fennec fox, as we can see here. So let's see, they say, fennec foxes live in the Sahara Desert, which is very hot. They have very large ears and a thin body. Remember, large ears, thin body, very hot. Explain how the body shape of a fennec fox has evolved by natural selection. So natural selection, or is any question asking you to reason in terms of natural selection, is requiring for the same thing. A mutation occurs, variation is created, and then survival for the fetus will go on after that. So I say a genetic mutation occurred in this, and it caused variation among fennec foxes. Foxes with large ears, as you can see, this one here has those large ears. So foxes with large ears and a thin body had a large surface area that enabled them to lose heat and stay cool in the desert heat. So this feature enabled them to survive in the desert heat, and they were able to reproduce. When they reproduced, they passed on their alleles or their genes to their offsprings, and over generations, these offsprings continued to survive, and then that is how they evolved to become the way they are. This brings us to the end of question 1. Let us go into question 2. Question 2, the diagram shows the structure of a human eye. This is the human eye we were given. I tried to label some of the parts like the cornea, the conjunctiva, the pupil, the iris, ciliary muscle, the suspensory ligament, as well as the retina. So the first question says, which of these is the structure labeled X? As you can see up here, X is the cornea. So I labeled that to be the cornea. The next part says, when looking at a close object, which row of the table shows the state of the ciliary muscles and the suspensory ligament? When you look at an object that is nearby, the ciliary muscles are going to contract, the suspensory ligaments are going to loosen, and when they do that, the lens is going to get an appropriate shape into an appropriate shape so that the light is bent the right way in order for it to be focused onto the retina so that you can see clearly. I will go to the next page. They say, explain how structure Y controls the light entering the eye when someone walks into the dark room. I want to take you back to structure Y. As you can see, structure Y is the iris. How does it ensure that when you enter into a dark room, light entering the eye is going to be controlled? So let's see, I said, when someone walks into the dark room, the pupil dilates. The pupil, okay, let me go back to the iris. Sorry, just a little bit smaller. The iris has two sets of muscles. We have what we call the saccular muscles, and we have the radial muscles. So when you enter into a dark room, you expect that the pupil has to dilate. In order for the pupil to dilate, remember the pupil is that hole inside your eye. For it to dilate, it means the radial muscles have to contract in order to pull away. And to do that, the saccular muscles have to relax. So these saccular muscles within your iris have to relax while the radial muscles do contract in order to allow the pupil size to become wider or to dilate so that you can be able to see clearly. So I will continue to the next part. The next part says, multiple sclerosis is a condition that can slow down the speed at which electrical impulses travel along neurons. The time taken for the blink reflex to occur can be used to help diagnose if someone has multiple sclerosis. The blink reflex causes the eyelids to close. Air is blown onto the eye, and the time taken for the eyelid to close is recorded. So they say the diagram shows the reflex pathway. So here we have the eye, air is blown. So here the information goes through the sensory neuron, and then to the relay neuron, and back to the motor neuron, which causes you to blink. So continuing, the speed the impulse moves along the reflex arc consisting of all three neurons in a person without multiple sclerosis is 77 meters per second. The time taken for the blink reflex to occur in a person with multiple sclerosis is 0.05 seconds. 0.005 seconds. Let me cut out that. They say the total length of the neurons in the reflex arc for the person with multiple sclerosis is that. So this is the length of the neuron. And then they say calculate the difference between the speed of impulse for the person with multiple sclerosis and for the person without multiple sclerosis in meters per second. So I first began by converting the distance here. They gave us 25 centimeters into meters. It's going to be 0.25 meters. And then I went on to calculate the speed of impulse in a person with multiple sclerosis. This is going to be distance divided by time. The distance moved is 0.25 meters, and the time is that 0.0050 seconds or 05 seconds. When we divide this, we get 50 meters per second. And again, they wanted us to find in meters per second, so this is what we get here. Now here, remember they gave us this speed as well, so the difference in speed is going to be this 77 minus the 50 we have calculated here, giving us a 27 meters per second as the answer. Let's continue. So here they say the speed of an impulse along the axon of the motor neuron for someone with multiple sclerosis is 120 meters per second. Suggest why the speed of the impulse calculated along all three neurons is less than the speed of the impulse along only one motor neuron. Now, there are synapses, and depending on which neuron, it can take longer or it can take shorter for these impulses to be transported to the next neuron or through the specific neuron they are going through. So I said because there are more synapses before motor neurons. Before the motor neuron, there are more synapses. It takes time for the neurotransmitter to diffuse. And again, remember at the synapse, we have one specific neuron and another specific neuron. And then maybe if the impulse is coming from this side, it takes some time for the neurotransmitter chemical to come into the synapse, and then for that to link onto the receptors onto the other side so that an impulse can be generated in the next neuron. So it takes some time. So the time taken for the neurotransmitter to diffuse through the synapse and for an impulse to be initiated in the second neuron could take longer, which could be different in just one motor neuron. So this brings us to the end of this question 2. I hope the explanation makes sense to you. Let us continue to question 3. Question 3, the diagram shows a yeast cell. So this is the yeast cell they've given us. We see the cytoplasm, the vacuole, the mitochondria, the cell membrane, cell wall, nucleus, and so on. There is a ribosome as well. So they say, in which of the labeled structures in the yeast cell does aerobic respiration occur? Of course, aerobic respiration will occur inside the mitochondria. The nucleus is for DNA replication as well as transcription. Ribosomes are for protein synthesis, and vacuoles will store solids or store substances. So it's only the mitochondria. The next part says, what substance is the yeast cell wall made of? The yeast cell wall is made out of chitin. Some people call it chitin, others call it chitin. It's made out of chitin. Cellulose is for plants. This is in animals, and that can be in starch. And so it's only chitin. So next part says, which of these structures found in the yeast cell would also be present in a prokaryotic cell? This is going to be a cell membrane because prokaryotic cells have no nucleus, as you can see I wrote here. Other organelles, however, they have a cell membrane. So I went with part A. Let's continue. So here they say yeast cells are often used when making bread. A student uses this method to investigate the effect of temperature on the height that bread dough rises. They place dough containing flour, sugar, water, and yeast into a measuring cylinder, and then they measure the height of the dough. They place the measuring cylinder in a 25-degree water bath, measure the height of the dough after 2 hours, and then repeat the experiment at temperatures of 35 and 65. So you can see here 25, then maybe 35, it's going to be faster, and at 65, due to the denaturation of the enzymes, that could be affected. So here we have the experimental setup. There is the measuring cylinder, there is the dough, there is the water bath to maintain the temperature, and then the thermometer to ensure that the temperature is as required. So they say the table shows the students' results. So we see the temperature variation, 25, 35, 65, and then the height of the start, they are all at 25. And we can see with the 25, there is the height is kind of good after 2 hours, but the 35 degree gives us the longest or highest height, while the 65 gives us the least. You can see this is because of denaturation. This is because the temperature was not that high enough. It was not optimum yet, and this is maybe closest to optimum. So we had the highest height. And here they showed us the percentage increase in height. Here we see a 40%, 80%, and an 8%. So that is it. So here they asked us to calculate that. I said increase in height is going to be, we can see here it's going to be 45 minus 25. That is the change in height. Now, the percentage increase will be change divided by the original times 100, which gives us 80%. I will continue to the next part. Explain why yeast causes the bread dough to increase in height. As you can see, yeast is going to carry out respiration, and respiration, because it's going to be aerobic, it leads to the production of carbon dioxide. The carbon dioxide bubbles are going to escape as the dough rises, or there will be the gas is going to be trapped in the dough as it rises. So the entrapment of the gas or CO2 gas into the dough causes it to rise. So if there will be some heating, then the bubbles will be escaping, so the creating the tiny holes that will be present in the bread. However, that wasn't required here. They were just concerned about the rising. So you can just say carbon dioxide gas is going to be trapped in the dough, causing it to rise. The next part says, explain why the dough rises to a different height at 25 degrees Celsius compared with at 35. So at 25 degrees, the kinetic energy is lower than that at 35. This is a fact. So we will have less collision between the enzymes and the substrate. And again, for any enzyme catalyzed reaction to occur, we need a collision between the enzymes and substrate so that they can combine in order for a reaction to occur and products to be formed. So this low collision between the enzyme and substrate will lead to less CO2 being produced, resulting in lower height than at 35 degrees Celsius. The next part says, explain why the dough rises to a different height at 35 compared with that at 65. Now at 65, because this is an enzyme controlled reaction, at 65 it's beyond the optimum. The enzymes are being denatured, so the substrate or the enzyme's active sites may be deformed or not having the same shape as they had previously, so the substrate might not be able to fit into those active sites, and therefore a reaction will not occur, so respiration is going to decrease and the CO2 produced is going to be lower, so the increase in the height is going to be really, really insignificant or kind of so low. So here I say at 65 degrees, enzymes are denatured or they are denaturing, so the substrate cannot fit into the enzyme's active sites. Remember, denaturation causes the enzyme active sites to be deformed or to be changed, so respiration decreases and less CO2 is produced, so there will be less increase in the height. This brings us to the end of question 3. Let's move on to question 4. Question 4, the diagram shows the human alimentary canal. So this is kind of the digestive system being shown here. They say, explain how food passes down the esophagus. Now the esophagus is around here, so how does food pass down? Food is going to pass down the esophagus by peristalsis. Peristalsis is the rhythmic movement of food, which is going to be created by the movement or the contraction of the basically the muscles within the gut. Then you might contract, pushing or squeezing the food, like toothpaste is squeezed out of its tube in order for the food to move through the gut. So I said peristalsis is created by waves of muscle contractions of the circular muscles of the gut. So this causes the food to move through the esophagus. The next part says, which labeled structure produces bile? Bile is going to be produced by the liver, and as you can see, the liver is C, which is the liver C. Continuing on to the next part, they say, describe the role of bile in digestion. Now, bile has two uses. Number one, it is to neutralize the pH of the acidic chyme or the food. The food leaving the stomach is acidic. It has to be neutralized by bile in order for the enzymes that are released in the duodenum to not be affected or to have the right pH for them to do the activity. The next thing that bile does is it emulsifies the fats in order to create a larger surface area so that when the enzyme lipase is acting onto these broken down particles or the emulsified fat, its rate of reaction is going to be faster. So I say bile neutralizes the acidic chyme. It therefore provides the optimum pH for the enzyme activity. The enzymes I'm talking about are the enzymes from the pancreas as well as those from the walls of the small intestine. Then I also said bile also emulsifies fat. This is breaking down the fat into small droplets, providing a large surface area for lipase action. So that is the purpose of bile. Lipase inhibitors are chemicals that bind to lipase enzymes. To test the effect of a lipase inhibitor, equal masses of full-fat milk are placed into 2 test tubes. Lipase inhibitor is added to 1 test tube. Lipase is added to both test tubes, and the pH of each solution is measured every 5 minutes. The results are shown in the table. So here in the table we have the results, and we can see as time passes by, here we have a greater decrease in pH in comparison to that. So the first question is asking, calculate the mean rate of pH change per minute of the solution without lipase inhibitor. So we are looking at this solution here. So here we have the starting pH and the final pH. So the change in pH is going to be 8.0 minus 5.8 divided by 20 because overall time. And again, remember they asked you for rate. Rate is any specific change divided by time. So since the time was 20 minutes, we divide by 20. and in the end we get 0.11. So the mean rate of pH change was 0.11 per minute. Explain the differences in the changes of pH of the solutions in the two test tubes during the 20-minute period. So here we see there is without lipase inhibitor, and in the other experiment there was with lipase inhibitor. Remember, lipase causes the breakdown of fats into fatty acids and glycerol. So if there is no inhibitor, the lipase is going to cause more fatty acid production, and therefore the pH is going to decrease faster. So I said without lipase inhibitor, the pH changes faster, meaning it's going to be more acidic, and this is because the fats are broken down into fatty acids, or they're broken down more to produce fatty acids and glycerol, and the fatty acid leads to pH decrease. Now, in the other experiment with the lipase inhibitor, there will be less fatty acid production, and therefore there will be less decrease in pH. Let's continue to the next part. So here they said doctors use this method to investigate the use of lipase inhibitor as treatment for obesity. They give 3 volunteers a tablet containing the lipase inhibitor and give another 3 volunteers a tablet with no lipase inhibitor. They give all volunteers 100 centimeters cubed of olive oil to drink, and then they measure the lipid concentration in the blood of the volunteers after 3 hours. Some of the volunteers reported abdominal pain 3 hours after drinking the olive oil. The part says the table shows the doctor's results. So these are the results of the doctors. They saw those who had abdominal pain or not, and we can see the blood lipid concentration at the start as well as after 3 hours. So let's continue to the question. So discuss the use of lipase inhibitor as treatment for obesity. Remember, this is for treating obesity, and use the data from the table to give your answer. So here I began by talking about the things where it's beneficial and then where we are skeptical about the experiment. So I said, lipase inhibitor prevents lipase from being broken down into fatty acids and glycerol. And if we do not have fatty acids and glycerol, then we cannot have absorption of these substances. So if there is less of fatty acids and glycerol, there will be less absorption of the fat. And if there is less, then there will be lower blood lipid, and there will be less deposition of these fats into the fat storage parts of the body, leading to less obesity. So this is beneficial. Now, on the other hand, the sample size was small. Remember, in every experiment, they used only 3 people. By experiment, they gave 3 people the lipase inhibitor and the others with not. So it was a very small sample size. And since some people experience side effects like stomach aches, this shows that the method may not be usable in real life because maybe some people will have reaction towards it. And then there was no data about the age of the people or the volunteers, their body size, as well as their sex. This is very important because people of different body sizes, different ages, as well as different sexes have different metabolic rates, and that could cause different results if they were experimented on. And there is no information about their diet, what do they eat, how much of that, do they frequently eat fat or not. We needed to know that information. And there is also no information about the starting blood lipid levels of the people. So all this information would be very important if we want to evaluate to see if this method is efficient or if it can be used properly. So this brings us to the end of this question 4 and the end of this video. I will put the links to part 2 as well as part 3 in the description box. Thank you for being with us. Please do not forget to subscribe. See you in the next video. Bye-bye.